Hi students, we have to discuss the last part of the lesson reproductive health. What is that? Detection of fetal disorders during early pregnancy. What are the different methods adapted to find out is there any defects in the fetus during the early pregnancy period or during what is called the gestation period. Now the first method, ultrasound scanning. This is a method adapted nowadays to find out the nature of the baby, the dating, number of fetuses, the heartbeat rate, etc. Not for heartbeat rate, we are using fetoscope for just finding out the heartbeat rate. But here we have to find out the complications during the early pregnancy, the number of fetuses, etc. So ultrasound has no what is called known risk, except that a mild discomfort, the one which is caused due to the pressure created by what is called the transducer, the one which transmits the impulses, that is called a transducer that is being placed on the abdomen of the pregnant woman. That is called a transducer, and it is being placed either on the abdomen or you see that one on the vagina to find out any abnormalities in the case of a child. So in the case of ultrasound scanning, there is no radiation procedure involved. So no radiations are required during this procedure. Now what is its importance? So it is usually performed during the first trimester up to 12 weeks for dating to determine the number of fetuses, either only one individual or twins are there, either fraternal or identical twins. And also for assessment of early pregnancy complications. Okay, so for dating how many days the embryo old, and then we have determination of number of, that is fetuses, and for assessment of early pregnancy complications. We have several types of ultrasound imaging techniques, 2D, 3D and 4D. Out of these three, 2D, 3D and 4D, 2D is the most common type. Now normally it provides a flat structure of one aspect of the baby. So flat structure of one aspect of the baby, that's in only one plane. That is called what we can say 2D ultrasound image. The next one, 3D ultrasound image. This allows the what is called the healthcare provider to see the height, the width and the depth of what we have the fetuses, and also that will be helpful in diagnosis of what is called the fetus during the development. So 3D, you know that one, the height, depth, width all being known, the image shows these three features, and that will be helpful in early diagnosis of what is called the fetus. Is there any complications? Now the third type is 4D. The third type is 4D ultrasound image. This is a modern technology. It helps the healthcare provider to visualize unborn baby that is moving, that is unborn baby moving in a real time. So within a short period, how does the baby move from one place to another? That is within this what is called the amniotic sac, along with 3D images. So the 4D normally provides information about or just helps the healthcare provider, the one who is observing, to see or to visualize the unborn baby moving in just a period of time, moving in a period of time, real time moment within a short period, along with 3D images. That is about the ultrasound scanning method, the three types, 2D, 3D and 4D, mainly for finding the early complications of pregnancy, number of fetuses, and also we can find out what's the dating, how long the embryo old, that is how long the embryo old, in terms of weeks and in terms of days. That's about ultrasound scanning, the first procedure. The second procedure we have to go, that is amino synthesis. Now the second method, amino synthesis, this is the oldest method, not a new method, but adapted somehow to find out any chromosomal abnormalities in the developing fetus. Now it involves actually taking a small sample of amniotic fluid. Amniotic fluid is the one which is being enclosed by the amniotic what is called membrane or amnion. So the amnion, one of the extraamniotic membrane, encloses the baby with a cavity. That cavity is filled with what is called amniotic fluid that surrounds the baby. So this method involves taking a small sample of the amniotic fluid, the one that surrounds, that is the fetus. The results are then analyzed to diagnose is there any chromosomal abnormalities like Down syndrome, Foetal syndrome or any other syndrome. Then this procedure is generally normally done during what is called, actually in the case of pregnant women only we are doing that one, between 15th to 25th week of pregnancy. What are they doing? By inserting a long thin needle, that is what is called hypodermic syringe, through the abdomen into the amniotic sac where the fluid is there. So the fluid is being withdrawn in a small sample and that is being analyzed. In that analysis we can find out not only the chromosomal abnormalities but also we can find out the nature of the sex of the baby, whether it is male or female. That is why there is a statutory ban on amniocentesis, because people are misusing that one to have what is called the female feticide. That's why it's being normally banned. So only legal medical termination is done, but amniocentesis is now banned. No one can do that one without any what is called legal opinion from the government. Okay, now, so why we are taking the amniotic fluid? Because the amniotic fluid contains the cells, and those cells are shed from what is called the fetus. Water cells we have in the fetus also found floating in the amniotic fluid regarding the male and female baby with the XX chromosomes and the XY chromosomes. Or we have the Down syndrome, Foetal syndrome, Klinefelter syndrome. Any syndrome can be seen with the help of what is called the analysis of the cells found in the amniotic fluid. That procedure is called amniocentesis. We have a beautiful picture. I'll show that one in the video. You can see that one. Okay, that's about amniocentesis. So we have to go for the next one, chorionic villi sampling. Now the third method of detecting the fetal disorders during early pregnancy is chorionic villi sampling. Chorionic villi, nothing but the component of placenta. So it is nothing but taking the tissue of placenta to test for chromosomal abnormalities. That's the meaning for that one. So it is a prenatal test just before what is called the birth of the baby. That involves taking a sample of placental tissue, because chorionic villi is a part of the placenta, to test for chromosomal abnormalities just like amniocentesis. Now what is fetoscope? Normally it is used to monitor the fetal heartbeat rate during the late pregnancy as well as during the labor. Now the rate of heartbeat in the fetus is always higher than that of a normal person. In a normal woman or a man, you have 72 beats per minute. But in the case of fetus, the rate of heartbeat ranges from, that is lying between 120 to 160 beats per minute. Suppose if you have an abnormal heart rate and there is a change in pattern of heart rate, it means that normally the fetus is just without getting enough oxygen. So the fetus is not getting enough oxygen and it indicates other complications or other problems to the fetus. So fetoscope is normally used to monitor the heart rate. Okay, this is with reference to monitor the fetal heart rate and other functions, mainly heart rate. Now we have a device what's called Doppler device. It is a handheld Doppler device. It is used to normally count the number of heartbeats in the fetus. Whenever during prenatal visits to a doctor, the doctor is checking the heartbeat rate of the fetus by using the Doppler. Okay, so the fetoscope, just normal fetoscope is like this one, the oldest one, in the form of plastic or in the form of metal or in the form of wood. They are keeping this one just like a funnel when it's being placed over the abdomen of the pregnant woman, and they are placing what is the earlobe to hear the sound. That's the oldest method. Now we have electronic fetoscope is available. I'll show you the what is called that is in the video. You can see that one, the various devices. I'll show that one. It's not easy to draw that one. So likewise Doppler device. It is also just we are keeping the transducer on the surface of what we call the abdomen, or you can find out the heartbeat rate in what is called actually the foot on the upper surface. That is a device often used to count the number of heartbeats in the case of fetus. Also we have electronic monitoring that is being done continuously during what is called the labor, during labor or during childbirth. So we have during labor a continuous electronic fetal monitoring is often used, whether the fetus is normal, whether the birth is going to take place normally or not like that. So Doppler device as well as fetoscope meant for hearing the heartbeat sound, counting the number of heartbeat in the case of fetus, whether it is normal or abnormal. That's about what we have the different methods adapted for detecting the fetal disorders during pregnancy. I want to give some facts, for example, some syndrome, what is cryopreservation, and also what is the role of vitamin, who has observed, that is the steroidal sex hormones, etc. Now what is Mayer-Rokitansky syndrome? Normally, generally, all women are born with ovaries. But some women do not have functional uterus. This condition is called Mayer-Rokitansky syndrome. So, do not have functional uterus. Absence of functional uterus. That condition is called actually Rokitansky syndrome. Now, what is cryopreservation? So, it is a method of freezing embryos. When there are more embryos actually present than needed for a single IVF treatment, the extra embryos are stored under frozen condition under minus 196 degrees Celsius using liquid nitrogen. This method is called cryopreservation. That is, preserving the embryos, even the semen or even we have the ova in a frozen state is called cryopreservation. Now what is the advantage of this cryopreservation? So embryo cryopreservation provide normally an additional opportunity to pregnancy, provide an additional opportunity to pregnancy, and that method is called frozen embryo transfer, FET. So embryo cryopreservation provides an additional what's called opportunity to pregnancy through frozen embryo transfer, without undergoing another stimulation of the ovary and also egg retrieval. We don't want to stimulate the ovary once again to retrieve the eggs from that ovary. That's the importance of what is called cryopreservation. So this is an example for what is called ex situ conservation. Ex situ conservation means not being conserved in the place where they are present, they are being taken away and conserved. Now some facts related to what we have the reproductive health. Number one, vitamin E. Vitamin E is aptly called anti-sterility vitamin. This is called anti-sterility vitamin. The reason for that one, it helps in the development and also normal functioning of reproductive structures. Number two, sex hormones were first discovered by Adolf Buchanan. Adolf Buchanan was a man just could discover the sex hormones. I want to give some more four factors, some facts related to reproductive health. Some other facts related to reproductive health. Now, syphilis and gonorrhea, both STD, sexually transmitted diseases, are commonly called international diseases because they are affecting all groups of people in different parts of the world. Then 11th July is observed as World Population Day. Then we have 1st December is observed as World AIDS Day. Now what is NACO? National AIDS Control Organization That organization was established in 1992. So with this we concluded the lesson reproductive health. We have to go to the next lesson in the next class. Okay, thank you.